Hello everyone, Dr. A again. I'm here with your lesson on the immune system. This is going to be part one of a two-part series. And we are again going to start with a case. So this is a 68-year-old patient with Alzheimer's disease. She was brought to the ER by the staff of a local nursing home. So she is a nursing home patient. I'm sorry, he sorry about that. He presented as lethargic with a sallow complexion. He had an admission temperature of 102.4 Fahrenheit and a respiratory rate of 33 per minute, which both are high. During respiration, the right side of his chest moved better than the left. He showed dense consolidation of the lower lobe of the left lung on physical exam. A sputum sample revealed blood in a greenish color, so he has green blood-tinged sputum. A chest x-ray showed tight consolidation of the left lung with evidence of a formation of cavities in the lung tissue from cytotoxic damage. The patient complained of chills in the exam room combined with his fever. A smear of his sputum demonstrated no acid-fast bacteria, that means not tuberculosis basically, but showed gram-negative, heavily encapsulated rods. He was diagnosed with bacterial pneumonia caused by Klebsiella. Cytotoxic damage to the lungs is classic with this type of infection. Okay, so let's first talk about pathogenicity. So pathogenicity is an organism's potential to cause infection or disease. So pathogens, pathos meaning disease, pathogens can generate disease. A true pathogen is capable of causing disease in healthy persons with normal immune defenses. They are generally associated with a specific recognizable disease. They can vary in severity from mild to severe to fatal, depending on the person, right? And examples include the influenza virus, the plague bacillus, and the malaria protozoan. So, for example, my friend gauged that, and he's a missionary, and he had malaria. Well, he was otherwise a very healthy young man, and maybe he's middle age now because he's about the same age as I am. And when he got malaria, he got sick. So that is a good example of a true pathogen. And opportunistic pathogens, however, they cause disease when the host defenses are compromised or when they become established in a part of the body that is not natural to them. They are not considered pathogenic to a normal, healthy person. They do not possess well-developed virulence properties, so things that make them able to really evade the immune system and establish infections. And some of these examples include Pseudomonas species and Candida albicans. So one of the cases we had looked at before, where the lady that had strep throat ended up with a yeast infection, vaginal yeast infection caused by Candida albicans. Well, in that instance, Candida worked as an opportunistic pathogen. It's actually normally present in the vagina. It just was able to grow, more because it wasn't inhibited by the bacteria, because the bacteria had gotten killed by the antibiotic. Also, so normally Candida albicans, everybody has some, but if you get a patient that is getting chemotherapy for cancer and stuff, and they're knocking out their immune system, they can very well get a Candida infection that's even systemic. So that happened to my aunt when she was being treated for acute myelocytic leukemia, pending her bone marrow transplant. They had knocked down her bone marrow completely, knocked it out. She had like no immune system, and she ended up with an infection, a fungal infection in her liver, in her lung, like everywhere. She was systemic, and she was in ICU for a while. She almost died. She thankfully did recover and was able to get her bone marrow transplant at that time. Okay, so in our case, do you think Klebsiella is a true pathogen or an opportunistic pathogen? So what do you think? All right, let's talk a little bit about healthcare associated infections. So in our case, our patient was a nursing home patient. Therefore, this infection that he got is because he lives in a nursing home, could be considered a healthcare associated infection. So even though generally they are considered infections that are acquired or developed during the hospital stay, if they're acquired or developed during a nursing home stay, that can also qualify as healthcare associated infections. They range from 0.1% to 20% of all admitted patients, with an average of 5% nationwide. So you want to know how good a facility is, find out their rate of healthcare associated infections. They are anywhere from 2 to 4 million cases a year that result in 90,000 deaths. So that is quite a serious thing for those 90,000 people that have died from it, obviously. And this is something that we can play a role in preventing as healthcare professionals. It causes 8 million additional days of hospitalization and an increased healthcare costs of $5 to $10 billion a year. That's a lot. The factors that are tied to healthcare-associated infections are compromised patients, so who goes to the hospital, sick people. It's collection point for pathogens, so you have a whole bunch of sick people in one location. And then the lower defenses, because of them being sick, can permit normal biota to enter the body, or normal biota that normally wouldn't cause problems to cause infections. And then infections can be acquired directly or indirectly from fomites, medical equipment, other patients, medical personnel, visitors, air, and water. So on the medical personnel, that would be, for example, why it's important for you to wash your hands. Because if you don't wash your hands, if you go from one patient's room to the next without washing your hands between each patient, you could carry bacteria or viruses and stuff on your hands and spread them from one patient to the next. And fomites, we're going to look at are things, inanimate objects, so things as doorknobs and all of that. So just to give you a little scenario, think of a patient that's in isolation for C. diff, and I know I've personally seen this. So you have all these isolation precautions. You've got to get gowned up, gloved up, masked up, and all of that stuff to just enter in the room and all of that. And then you have a visitor who's gone into that room, has not gowned up or gloved up or done any of the stuff. They've been in the room, they've touched stuff, they leave the room, they don't wash their hands and stuff, and then they go and use a water fountain. So now the little water fountain knob is contaminated. They push the elevator knob. Now the elevator knob is contaminated. They maybe go get a soda somewhere. Now the vending machine is contaminated and stuff. So anyway, some of the most contaminated places that you can find are these knobs and buttons and vending machine buttons and microwave buttons and these kinds of things, because we don't think about washing those and things. And so even think in the age now with coronavirus, with they're putting these shields up for when you check out and all that to limit the spread. But those pin pads where you put in your code for your card and all of that, they're not wiping those down. Those are not being decontaminated. So, and everybody is touching them. Anyway, so that would be, you could get it that way through fomites and stuff. Okay, so the most common healthcare associated infections are, this is in order, so we have urinary tract, respiratory tract, and surgical incisions infection. And so if you can see here in red is the urinary tract, in green is the surgical site, and in orange are the respiratory infections. So just that is 3/4 of the hospital or healthcare associated infections. Then next is gram-negative intestinal biota. So E. coli, Klebsiella, and Pseudomonas. So ding, 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 Klebsiella, that was our patient's case, right? These are cultured in more than half of the patients with nosocomial infections. So our patient had a respiratory infection from Klebsiella, and it's an intestinal, so think poop. So how could he have gotten it? For example, just think, how could a bacteria that is normally in feces end up in the respiratory tract of a nursing home patient? Okay, just think about that for a second. All right. There are also gram-positive bacteria, so staph and strep, and yeast. They make up the rest of the organisms that cause healthcare-associated infections. So true pathogens such as Mycobacterium tuberculosis, Salmonella, hepatitis B, and influenza can be transmitted as well. And so, yeah, if it is possible for especially flu and TB and all that to move through facilities, whether it be hospitals, but even schools, prisons, and that kinds of stuff. All right, so let's talk about now signs and symptoms of disease. So those signs and symptoms are warning signals of disease. So they're usually an indication that something is amiss in your body. It doesn't specifically have to be an infectious disease, but infectious disease certainly do have signs and symptoms. So it is important for you to make a distinction between sign and symptom. So a sign is an objective evidence of disease as noted by an observer. Okay, so if you as a healthcare professional or as a nurse can observe and chart that that evidence, then it's a sign. So for example, a fever that you can measure that has a specific number, that is a sign, right? A rash that you can see, a specific wound in a specific location or something like that. So those are signs. So you can yourself observe them. A symptom is subjective evidence of disease as sensed by the patient. So the patient has to report it, and you really have no way of verifying whether they're telling the truth or lying. So definitely pain is always a big symptom. Even like, I would say shortness of breath could be a symptom, even though a sign could be how many times a minute are they breathing, and you could notice labored breathing and stuff. But the feeling of being short of breath, it is also kind of a symptom. So pain is definitely a big one because it's totally subjective. People can lie like crazy. Nausea is another one that is very subjective because somebody may not look right, but do they have nausea or not? And if they're not able to communicate, you may not be able to know if that's what's going on with them. Okay, and then a syndrome is a disease identified by a certain complex of signs and symptoms. So think about if we say fever, fatigue, muscle aches and pains, sore throat, headache, coughing. Okay, so what would you think about that? So I would say this kind of syndrome with the fever and everything, I would automatically probably start thinking maybe flu. All right. If you said frequent urination with burning upon urination and stuff, and maybe even some fever and stuff, then maybe you think UTI, right? So those are syndromes. They're a cluster of signs and symptoms that indicate a certain disease and all that. So in this description of our case, I want you to underline all the signs of pneumonia. Okay, so you can highlight it. This is a little highlighter, so you can see it's a little fainter. So if you pick it, and you can again change the thickness, and then you can do stuff like that, right? So that's how you use the highlighter. Okay, so let's talk about portals of entry. So there are healthcare processes that can lead to healthcare-associated infections. So any treatment using reusable instruments, such as respirators or thermometers. So that's where you have the thermometer covers for the thermometers. But respirators have to be completely decontaminated between patients. And anytime you mess with it, there's chances of introducing infection and contamination into the patient. Any indwelling devices, such as catheters, prosthetic heart valves, grafts, drainage tubes, tracheostomy tubes, all form ready portals of entry. And a high proportion of the hospital population receives antimicrobial therapy. So drug-resistant microbes can be selected at a much higher rate because so many people are on antimicrobials or antibiotics. The portal of entry is the route that a microbe takes to enter the tissues of the body to initiate an infection. So there are two types of portals of entry. There's exogenous and endogenous. So exogenous is the microbe came from a source outside of the body, so from the environment or from another person or an animal. So think of maybe being in an elevator or something like that, and somebody sneezes, and then you breathe in their sneeze droplets. That would have been an exogenous portal of entry, and it came because it came from another person and from them sneezing. Endogenous is microbe already existing on or in the body. It's usually part of normal biota, or it was a previously silent infection. So C. diff is one of those examples, and the vaginal yeast infection is another one of those examples. So both of those organisms are normally present in lower amounts, but then antimicrobial treatments can knock down the competition and allow those those infections to take hold and to those organisms to just grow out of control. Okay, so typical portals of entries and organisms involved. So for the skin, we have Staph aureus, Strep pyogenes, Clostridium tetani, herpes simplex, helminth worms. They can enter through your skin, walking around barefooted and stuff like that. Viruses, rickettsiae, protozoans like malaria and West Nile virus because of mosquito bites and stuff. And Haemophilus, aegypticus, chlamydia, trachomatis, and Neisseria gonorrhea. And they can enter through the conjunctiva of the eye. So, helminth worms burrow through the skin. And of course, staph would enter through nicks and lacerations, abrasions, punctures, any, broken skin. Staph, strep, and Clostridium tetani can all enter through there. Herpes simplex, mucous membranes of the lips. So that's how you get the cold sores and stuff. Those mucous membranes, kissing somebody that has it. Gastrointestinal tract is another portal of entry. So that would be, through the mouth usually. So Salmonella, Shigella, Vibrio, E. coli, polio, hepatitis A, echovirus. By the way, norovirus too, rotavirus, and enteric protozoans such as Giardia lamblia and Entamoeba histolytica. And you get them by eating or drinking contaminated foods or fluids via fomites, which are inanimate objects contaminated with the infectious organism. And then, of course, you would then handle something like a piece of food or something and put it in your mouth. And then, so if you had contaminated hands, that's how you get it. The respiratory tract, so there would be inhalation of the offending organism, usually in cough droplets, and bacteria causing the meningitis, influenza, measles, mumps, rubella, chickenpox, common cold, strep pneumo, Klebsiella, mycoplasma, cryptococcus, pneumocystis, mycobacterium TB, and histoplasma are all inhaled into the lungs. And then your urogenital tract, so they're enter either through the skin mucosa of the penis, the external genitalia, vaginal cervix, urethra. They may enter through unbroken surfaces or through a cut or abrasion. So this is HIV, trichomonas, hepatitis B, syphilis, treponema pallidum, Neisseria, gonorrhea, chlamydia, trachomatis, herpes, and genital warts. Okay, so what portal of entry did our Klebsiella use in our case? So go back if you need to, arrow back to the case description. You can do that. Okay. Virulence. Let's talk about virulence. So the virulence is the relative severity of disease caused by a particular microbe. It's the degree of pathogenicity. The virulence of a microbe is determined by its ability to establish itself in a host and cause damage. Virulence factors are any characteristic or structure of the microbe that contributes to its ability to establish itself in the host and cause damage. All right, let's talk about infectious dose, because this is related to virulence. So an infectious dose is the minimum number of microbes necessary to cause an infection to proceed. Microorganisms that have small infectious doses have greater virulence, right? If it takes just a few to cause a disease, then it is, quite easy for it to establish infection. So the infectious dose for rickettsia, for example, is a single cell. You get that through tick bites. So one tick bite can give you rickettsia, which gives you Rocky Mountain spotted fever. The infectious dose for TB, gardaiosis and coccidioidomycosis. So Giardia, remember Giardia lamblia we looked at, and TB, you should know, is, so those are about 10 cells, so not a lot. And just to really freak you out, if somebody with TB coughs, cough, or even doesn't cover the droplets for TB, they can stay suspended. TB can stay suspended in the air for like 2 hours. 2 hours. So like the person can be done gone. You don't even know they were in there and coughing, and you could breathe it in and be exposed to TB. The infectious dose for gonorrhea is 1,000 cells. The infectious dose for typhoid fever is 10,000 cells, and the one for cholera is 1,000,000. So, which you think is a lot, but cholera is spread through contaminated drinking water, and so if you're drinking contaminated water, it's actually quite easy to get that many. And so, this diagram I really do like, because it can kind of give you this idea of who gets sick, who doesn't get sick. And this is actually quite pertinent to what we have going on right now with coronavirus. Okay, so let's start here. So if the virulence of the microbe is low, and you get a low percentage of the optimal infectious dose, which we don't know what the infectious dose is for coronavirus or anything. We don't know how many virions it has to be. So it's not very virulent. Let's say this organism is not very virulent. You don't get a big dose. It's not the correct portal of entry, right? So maybe you had gotten it on your hands and you washed your hands and it doesn't come through the skin or anything like that. So these are all factors that are related to the microbe itself. And then these factors are going to be related to the host. So a genetic profile that resists the microbes. So those are non-specific defenses. So maybe you have really good intact skin. Maybe, for example, there are some people in Africa that have certain antigen on their red cells that prevents the malaria organism from getting into the red cell. These guys are resistant, genetically resistant to getting malaria. And then another factor from the host, you've had a previous exposure to the microbe, so you actually have specific immunity, so your immune system knows what to do with it and can fight it off really well. And you're in good general health, okay? So if you have all of these factors here, the microbe can pass through completely undetected. You fight it off. You never know you have it. You never know it even came in contact with you. Okay, so now let's look at something. Virulence a little bit higher. You get a bigger dose of it, and it's the correct portal of entry. So maybe for our coronavirus, you got exposed to somebody that either coughed or talked or something, and you didn't have a mask on or whatever, and you breathed it in. Okay. But you have a good immune system. Maybe you were exposed to it previously, okay, and you've already recovered and you have a good immune system, right? So that would be my husband currently. He had coronavirus, but he's immune to it now. So even if he gets exposed here, and he's in good health, for him, the microbe more likely is going to pass through unnoticed, or it will become established without disease. So it's colonization without infection. So that can happen to people, and so they can actually have the virus or have the microbe, carry it, not know that they're sick, and so in a way they become those silent carriers. Okay. Or you can have, again, virulence is down a little bit and you don't get a big dose. It's not the correct portal of entry. You have good resistance. You have not been exposed to it previously, but you're in good health. It could pass through unnoticed, Or again, it could be established without causing disease. So wrong portal of entry, even if you've not been previously exposed to it, these make it where it's just probably going to pass through. You never know it even got on your body. And then here, if you have a high virulence organisms and you get a good dose of it in the correct portal of entry, your genetic profile is that resistant microbe is already down. Let's say maybe you have an autoimmune diseases or some of these other underlying conditions like diabetes that makes you just not able to fight it off real well. You've never seen it before. So that's some of the problem with coronavirus here. It's a novel virus, so nobody really has immunity to it, at least at the beginning. And your level of health is kind of like halfway there. Then this microbe can cause disease. So this illustrates like how really in every day we encounter probably a lot of microbes. There's a lot of stuff that's going to pass through our body and that actually just doesn't cause diseases or problems. And so if you follow stuff on social media, some of the posts, so many people have been emphasizing this, okay? Be healthy, take care of yourself, get adequate sleep, support your immune system, get good nutrition, take some multivitamins, all do that. So like manage your stress. If you have diabetes or something like that, manage your condition, do everything that you can to up this right here, wash your hands and all of that, so that if you come across it, you end up being one of those where it just passes through unnoticed. Okay, so establishing the infection. So if it has entered in you and is going to establish an infection, there are several things that have to happen. So the first one is adhesion. So it's the process by which the microbe will gain a more stable foothold on the host tissue. It could be from, the bacteria being able to stick to your tissues, to the virus being able to get in, and the receptors of the cell binding on the virus spikes. Okay. So it is dependent on binding between specific molecules on both the host and the pathogen. The pathogen is limited only to those cells and organisms to which it can bind. It has to be able to get in. So that's, again, with a virus, that's what makes them be able to infect certain cells and not others. And for bacteria and stuff like that, that's what allows them to usually infect, one body system versus another. A firm attachment is almost always a prerequisite for causing disease, since the body has so many mechanisms for flushing microbes from tissues. The adhesion mechanisms of microorganisms can be fimbriae and pili on bacteria, surface proteins, so again on bacteria, adhesive slimes or capsules, all those are on bacteria and stuff, and maybe amoebas and stuff like that. And then viruses can attach by specialized receptors. Worms and stuff can hook onto your skin. Yum, right? So thinking back about our case, which of these do you think Klebsiella used? So Klebsiella, again, is a bacteria. So would it be fimbriae, surface proteins, or adhesive slimes or capsules? So again, it's a gram-negative rod. It can be found on nose, mouth, and GI tract. And it's in the case description if you need to go back and refresh yourself on that. So, surviving the host defenses. So once it's able to actually stick and stay in, now it is going to be attacked. So it has to be able to survive host defenses. So phagocytes are cells that will engulf and destroy pathogens by means of enzymes and antimicrobial chemicals. So as illustrated right here, it's a cell that can come in and just eat the pathogen. But some pathogens can produce what we call antiphagocytic factors, so they can fight back. So antiphagocytic factors are virulence factors that help pathogens to avoid these phagocytes. So leukocidins, the leuko for white, cidin is to kill. They kill phagocytes, so they kill white cells outright. So slimes or capsules can do that, which makes it difficult for the phagocyte to engulf the pathogen. And an extracellular slime or capsule makes it difficult for it to engulf the microbes. And some bacteria, because of their, capsules and slime stuff, if they do get ingested, it can make it easier for the bacteria to survive inside the phagocyte. And so it actually is just, it just found a new home. And it can live there for a while and even duplicate itself. So, causing diseases. There are 3 ways that microorganisms can cause damage to their host. Directly through the action of enzymes, directly through the action of toxins, which could be endotoxin and exotoxins, which we'll talk about here in a second, or indirectly by inducing the host's defenses to respond excessively or inappropriately, and then it's actually your own immune system that's causing all the damage. So again, as we think back on coronavirus, some of the problems, some of the ones that are ending up on the ventilator. Really, the problem is an overreaction of the immune system, and that's what's causing them to be able to have trouble breathing and stuff. All right, so exoenzymes. So those are enzymes that are secreted by microbes, and they can break down and inflict damage on tissues. So enzymes usually can break stuff down, and so they just secrete so secrete them out into the tissue, and it can dissolve the host defense barriers, and it can promote the spread of disease to other tissue. Example of exoenzymes, we have mucinase, digest the protective coating on mucous membranes. Hyaluronidase will digest a ground substance that cements animal cells together, so it can actually separate your cells so it can get in. Coagulase can cause clotting of blood or plasma, and kinases can dissolve fibrin clots, which then can cause bleeding. Okay, so then another thing they can do is produce toxins, which are really a potent source of cellular damage. So it's a toxin is a specific chemical product of microbes, plants, and some animals that is poisonous to other organisms. So it is poisonous or toxic to other organisms. Toxins are named according to their targets. So neurotoxin act on the nervous system, enterotoxins act on the intestines, nephrotoxins damage the kidneys, and hemolysins disrupt the cytoplasmic membranes of red cells, making them lyse or rupture. An endotoxin is a lipopolysaccharide that is part of the outer membrane of gram-negative cell walls. So if you go back in the bacterial chapter, where we talk about the cell walls, the gram-negatives, remember you had the cell membrane, you had a thin peptidoglycan layer, and then you had another outer membrane that resembled the cell membrane, and it was made of lipopolysaccharide. Well, that outer layer there is an endotoxin. It has a variety of systemic effects on tissues and organs. It can cause fever, inflammation, hemorrhage, and diarrhea. And it's usually released when those gram-negative bacteria are lysed or burst, and then you have little pieces of their cell wall floating, and those act as toxins. So this illustrates each of those. So again, enzymes. So here, like the hyaluronidase, which can cause the cells to not stick together and bacteria to be able to get in between your cells. That is how, if you will, staph can cause really bad infections, even if you don't have an open wound or open skin, and then you touch something that has staph on it, and then it establishes an infection wherever you touch. It's because of that right there. It's because of it being able to dissolve your tight junction barriers here and actually get in and cause infections. And then your toxins, you have exotoxins and endotoxins. So exotoxins are toxins that are secreted by bacteria. So the tetanus toxin that's secreted by Clostridium tetani, that's a neurotoxin, that is an example of an exotoxin. And then the endotoxins are the cell wall of gram-negative bacteria, causes fever, makes you feel really like crap. And then an overreaction of the immune system or the host's defense. So you have like streptococcal organism or something like that, and it's in there and in the lungs, and your immune system is fighting against it and is producing a whole bunch of mucus and inflammation. Well, the mucus and inflammation is going to interfere with the exchange of oxygen and cause damage to the lungs and damage to the host. Okay, so going back to the case, which of these do you think caused the damage done by Klebsiella, which is gram-negative rod? An exoenzyme, an exotoxin, or an endotoxin? All right, the process of infection and disease. So then you have establishment, spread, and pathologic effects. Sorry. The microbes eventually settle into a particular target organ and then cause damage at the site. The host tissues are weakened as a result of the multiplication of the pathogen. The pathogens obstruct tubular structures such as blood vessels, lymphatic channels, fallopian tubes, and bile ducts. That's where you can get swelling and loss of functions and stuff like that. They can cause necrosis. So necrosis is accumulated damage due to pathogens that leads to cell and tissue death. So that is really not a good sign. You don't want your tissue dying. And viruses can destroy the cells by simply multiplying within the cells and then lysing the cells, which are rupturing the cells. And then you have the portals of exit. So all the ways that you basically can be infectious to other people, where bacteria can, or viruses or microorganisms that have established an infection, them can then exit your body and try to go cause infection elsewhere. So we have obviously through coughing and sneezing, and even sometimes talking and stuff, right? Through the skin, if you have again you're oozing out of a wound or something like that, right? Through urine, if your urine is infected. Through feces, if your feces is infected. Sexual organs too, if those are infected and you're having unprotected sex. Through blood. So if somebody removes a blood sample and that you have an infection in your blood, then obviously that could be a potential source of infection for someone. Through insect bites. So think about the protozoan chapter, through the life cycle of malaria. So how does malaria spread from one person to the next? Is through insect bites. So a mosquito can give the person malaria, and then once malaria is established, another mosquito can bite the person, pick up the malarial parasite, and then go bite another person and spread the malaria. Okay, let's talk about the stages of infections. So every organism has an incubation period, and the incubation period is the time it takes between the time you were exposed to the time you start getting some of the first symptoms. Okay, and again, going back to coronavirus, that was part of the problem, is we weren't sure what that incubation period was, and they've pretty much been saying anywhere from a couple of days to a couple of weeks, and that is why the quarantine time has been 2 weeks, because they've said it could be as long as 2 weeks. And so if you were exposed to somebody, you basically need to wait 2 weeks to see through the average incubation period to see whether or not you're going to move into this area here, the prodromal stage. So the prodromal stage is this general feeling that you're coming down with something. You don't feel good. You have the beginning signs and symptoms. Maybe you feel tired, run down. You just need a nap. You need to rest. Maybe you're starting to get achy or something like that. So that's the prodromal stage. Then as the symptoms intensify, then you're like, oh yeah, I'm for sure sick. You have you're at basically the period of invasion, and then the symptoms get worse and worse and worse until you get to the height of the infection, and then they kind of level off, right? And so my husband actually had coronavirus, and we think that his incubation period was only a couple of days, 2-3 days. And it was just one day he's like, ugh, I'm not feeling all that great. And it wasn't long until he went into this period, and then this whole period of invasion and height of infection lasted about a week. Okay. And then, felt like crap, fever, body aches. He just really wasn't feeling good. And then when the symptoms start to get better, So the fever started to go down, or the fever goes away, and then slowly the body aches are going away, and then you're regaining energy and all that. You're in the convalescent period right here. So, um, convalescent, you're recovering. So you're not back to normal yet, but you're recovering. So, that one, and it didn't take too long for him through the convalescent period. And then once you have recovered all your energy, you're completely recovered, then you're just back to normal. So those are the stages of an infection. And it is important to know that some infections can spread at different stages. So obviously during the height of an infection, it is likely that an infection could be easily spread to another person. But there are some infections, such as the coronavirus, which can transmit during the incubation period when you have no symptoms and you don't know you have it. Obviously, also, you could transmit it then through the prodromal stage, and you could transmit it again through the convalescent period. So this was something that was really interesting that we found out with my husband. His hospital required, one of the hospitals that he works for, the main one, required that he have 2 negative tests for coronavirus, not the antibody test, the one with the nose where they do the viral PCR one, and before he could come back to work. And we were like, okay, so once we had met all the requirements of so many days post fever and all of that from the CDC and Arkansas Department of Health, and so we were somewhere in here, we were like, okay, let's go ahead and let's order the first test and get it back, because it takes a few days to get it back, right? And then once we got it back, we could order the next one. Well, we ordered the first one, and a few days later it comes back. He's still positive, and he's feeling fine. He has like 0 symptoms, no fever, totally fine. So he obviously had still stayed at home and all that, but he was definitely a potential source of infection for other people. And then, so they're like, no, maybe wait until next week and then we do the other one. So we waited a week. So a week from the time we did the first test and did a second test, and that one ended up being what they call indeterminate. And then we did one a few days later, and that one ended up being negative. So basically, for at least an entire week after his signs and symptoms were gone, his fever was gone, over a week later, he was still positive for coronavirus and could have spread the infection. Okay. Now let's tackle the host defenses. So you have 2 main branches of your defenses of your immune system. You have the innate immune system, which is very nonspecific, and then you have the acquired or specific immune response. Within the innate nonspecific, you have what we call our first line of defense and our second line of defense, and then the acquired and specific is always the third line of defense. So let's quickly explore here. So innate, just think of built in. Okay, they're a part of you. They're a part of who you are. So the first line of defense are surface protection composed of anatomical and physiological barriers that keep the microbes from penetrating sterile body compartments. You have physical barriers and chemical barriers. So for example, intact skin, unbroken intact skin is a good physical barrier. That's the first line of defense. Okay. A chemical barrier is there's a lot of enzymes and stuff, for example, in your tears, in saliva, in mucus and stuff that are there to attack and break down any kind of bacteria that would get in those areas. Okay. Second line of defense are cellular, so there's some cells here, and chemical systems that come immediately into play. And so that's the idea, is they're the first responders to an infection. And if an infectious agent has made it past your first line of defense. Okay. Within those, you have your phagocytes and phagocytosis, which we're going to talk about. You have inflammation, you have fever, and you have antimicrobial proteins. Okay, so the organism has made it in, and these are the security crew and security things that are put in place. I am going to note there are gamma delta T cells and natural killer T cells. These guys are a bridge between the innate and the specific. So these guys are quite special. They're fun to go study. I've done some research on them, but they can act quickly and non-specifically, but then they can also get modified and become specific. So they can work in both capacities. So it's really interesting here. And then your third line of defense is they include specific host defenses that have to be developed. They're unique for each microbe. And they're through the action of specialized white cells, which are lymphocytes, and their B and T cells are involved in that. And this is what causes you to produce antibodies, and the antibodies are very specific. Okay, so for example, we're talking about coronavirus in antibody testing. If you have been exposed to coronavirus, they should be able to detect antibodies in your blood that you have formed to coronavirus. If you have never been exposed to it and never fought it off, you wouldn't have any kind of antibodies to coronavirus. So, this whole process of forming antibodies is your acquired or specific defenses, and it takes time for this to happen. Okay, so let's look at first line of defense in a little bit more detail. So you have your sebaceous glands that are part of your skin, your tears that have lysozymes, you have mucus, you have saliva, which also have lysozymes, earwax, the low pH of the skin, sweat that pushes stuff off of your skin. You have cilia in your respiratory tract, which are illustrated here, which move microorganisms back up with the mucus and causes you to cough them up, right? So the mucus is, also mucus and cilia together in your respiratory tract. You have stomach acid that destroys anything that you swallow. You have intestinal enzymes that can break down stuff in your intestines, and there's also mucus in your intestines. Defecation, so pooping, is a way to get rid of stuff, waste and stuff that you don't want. Urination and intact skin are all first line of defense mechanisms. So your non-specific chemical defenses, you have, again, your sebaceous secretions do exert an antimicrobial effect. Specialized glands of the eyelids lubricate the conjunctiva with antimicrobial secretions, so those antimicrobial chemicals, right? The lysozymes are an enzyme found in tears and saliva that can hydrolyze the peptidoglycan cell wall of bacteria, so it can destroy bacteria. You have a high lactic acid and electrolyte content of sweats. Again, not favorable for bacteria and stuff. Acidic pH and fatty acid content of skin. You have hydrochloric acid in the stomach that gives protection against the microbes that are swallowed. You have digestive juices and bile in the intestine that are destructive to microbes. Semen has antimicrobial chemicals, and the vagina has a protective acidic pH maintained by normal flora. So those are all non-specific chemical defenses. Everybody got them, right? And it doesn't matter what comes in. That's part of what's there to help you. Now, your second line of defense are your generalized and non-specific defenses that support and interact with specific immune responses. So, phagocytosis, inflammation, which we're going to go into phagocytosis in detail here in just a second. Inflammation, which is the redness, swelling, and heat that you see around wounds and insect bites and stuff like that. We have fever, which is an increase in body temperature. We have antimicrobial proteins. So in those, we have interferon, complement, and antimicrobial peptides. We have pathogen-associated molecular patterns, which are PAMPs, which are markers that many different kinds of microbes have in common. And basically, they flag them as, pathogenic microbe or microbe that just doesn't belong and needs to be removed. And then you have pattern recognition receptors, or PRRs. They're used by host cells with important roles in the innate immunity, and they recognize the PAMPs. So they're receptors, antigens or proteins. The receptors are on the white cells, antigens and the proteins are on the microbe. They interact, and the microbe in the first line of defense recognizes the white cell in the first line of defense recognizes the microbe as a pathogen, and that's all it knows. It doesn't know what specific pathogen it is. It just knows it's a pathogen, so it must be destroyed. And then, of course, non-self proteins that are not harmful are generally recognized as such, and the immune system is signaled not to react. So those would be your non-self proteins, would be like your bacteria that are part of your flora and your microbiome that are supposed to be there. They're harmless. And these guys, so they, if you will, they have their own kind of like ID card, and the immune system recognizes, okay, you're not self, but you're not harmful, so you're good to go. Okay, so the immune system will, right here, the purple guy, will go and it checks cells that are self, and it should not be a reaction. If you have a reaction of the immune cell against self, then you're going to have autoimmune diseases. And then if it makes the immune cell makes contact with a foreign cell, like a bacteria, it uses its pathogen recognition receptors, and it interacts with the pathogen-associated molecular pattern molecules, and it sees it as a pathogen, it automatically is just going to eat it and destroy it. Okay. So other parts of the second line of defense, there are general activities of phagocytosis. So the phagocytes, they move around and survey the tissue compartments, and they discover microbes, particulate matter, which are just bits and pieces, if you will, in injured or dead cells. So things that just trash, if you will, things that just doesn't belong. And they will automatically ingest and eliminate those materials. So they're part of the cleanup / security crew. And they will extract immunogenic information from the microbes and use that to often activate the adaptive immune system. There are 3 types of phagocytes. They are your neutrophils, your monocytes, and your macrophages. Your neutrophils are mostly responsible for eating and killing bacteria and are what you can see in pus. Your monocytes are they're related to macrophages. Your monocytes will attack bacteria, viruses, fungi, dead cells, whatever. They're the most versatile, and when they're in your bloodstream, they're called monocytes. Once they move out of the bloodstream and they locate into tissues, they're called macrophages, but they have the same job. Okay, which line of defense are antimicrobial proteins like interferon? Are they first, second, or third? And which line of defense are the high lactic acid and electrolyte content of sweat? Is it first, second, or third? All right, so let's look at the divisions of the immune system and all of that. So we're going to start with a hematopoietic stem cell. So this is the stem cell in your bone marrow. Okay. There are 2 major branches, as you can see, 2 major division. All right. And so the first one is the lymphocytic one. And so the lymphocytes will ultimately be responsible for your adaptive or specific immunity. So you get a lymphoblast stem cell. So you have basically a lymphocyte stem cell there, or lymphocyte precursor. It will develop into here lymphoblasts, and then they will develop into they'll be like naive lymphocytes, and then they can develop into B cells and T cells. Okay, so you can read a little bit. We're gonna get a little bit more into that later, and then so you have B cells and T cells. B cells do antibodies. T cells look for infected cells or cells that do not belong. So cancer cells, viral infected cells, etc. And within the T cells, there are some subcategories. You have your natural killer T cells and your gamma delta T cells, which again can work as part of the innate immune system and as part of the adaptive immune system. So they can respond to those pathogen-associated molecular patterns, but they can also act in a very specific manner. And then one of the offshoot of the lymphoblasts are also some of just natural killer cells, and they're patrolling your body, again, looking for viral infected and cancer cells, but again, they're not specific. So they're part of the innate or non-specific defenses. Okay, so then you have this whole other division right here, which is kind of like the myelocytic division, if you will. So you have a stem cell, that comes off of your hematopoietic stem cell. And that stem cell, then, depending on the signals that the body sends to the bone marrow, can either develop into an erythroblast and make red cells. They can develop into a megakaryoblast and make a megakaryocyte, which makes platelets. They can develop into a myeloblast, okay, which can also it can go on and become a megakaryoblast if it needs to. But the myeloblast then can make the neutrophils, the basophils, and the eosinophils, okay? And it can also make mast cells. So mast cells are very similar to your basophils, but they're located in tissues, and they release histamine and stuff like that when you have allergic reactions and stuff. Okay, so all of those are related to this one. And then another branch of this stem cell is going to be the macroblast here, where you get your, the monoblast, where you get your monocytes and the macrophages. And the monocytes can also become dendritic cells. So macrophages and dendritic cells are like cousins. They have the same kind of job, and they both are located in tissues and stuff. So that kind of gives you a quick overview of all the players in the immune system. And then, so this right here is white cells, immune system. These two are your red cells and platelets, okay? But they all come off of this hematopoietic stem cell that's located in the bone marrow. Okay, so the mononuclear phagocyte system. Phagocytic cells are enmeshed in your reticulum, which is the support network of connective tissue fibers that permeates all the tissues of the body and interconnects nearby cells and meshes with the massive connective tissue that surrounds every organ. Think of this support network and all that as so it surrounds and holds tissues together and all of that, but it's also kind of like a highway for these cells to travel. The phagocytic cells are found in the lymphatic system, so this mononuclear phagocyte system. So we have the thymus is the site of blood cell maturation for the lymphocytes to become T cells. Lymph nodes have lymphocytes and macrophages and stuff. Tonsils, again, same thing. It has lymphocytes, macrophages, and stuff. The spleen, same thing, has lymphocytes and macrophages. Just the spleen filters blood, and whereas all the others filter lymph fluid. And then the lymphoid tissues of the mucosa of the gut and respiratory tract also are full of lymphocytes and macrophages and stuff. So this mononuclear phagocyte system is loaded with white cells that are called macrophages that are waiting to attack passing foreign intruders as they arrive in these locations. So your neutrophils are, again, general purpose phagocytes. They react early in an inflammatory response to bacteria. So if the neutrophils are increased in the patient's CBC report, then you can suspect a bacterial infection. They also respond to other foreign material in damaged tissue. Again, a high neutrophil count in the differential on the CBC is a very common sign of a bacterial infection, which is why ER physicians like to order CBCs, because they can at least they can help them determine whether the infection is going to be bacterial in nature or viral in nature. And again, neutrophils are the primary component of pus. Monocytes and macrophages, so they're the kings of the phagocytes. So monocytes are, again, transformed into macrophages after they migrate out of the bloodstream and into the tissues. A histiocyte is a macrophage that lives in a certain tissue and remains there during their lifespan. So they're a tissue-bound macrophage. So basically, and then they can have specialized names. So in the lung, they're alveolar macrophages. In the liver, they are called Knupfer cells. In the skin, they're called dendritic cells. And the macrophages in the spleen, lymph nodes, bone marrow, and kidney, bones, and brain are also histiocytes. So they, basically once they home to a certain area, the histiocytes, their macrophages, they stay put. They, if you will, they specialize in guarding that area. And phagocyte just means eating cell. In phagocytosis, is the physical process of the engulfment of that particle and the attack and dismantling of the foreign cells. Okay, a macrophage would be effective against which one of these? So select which one you think is correct. And let's look at phagocytosis. So one, you have chemotaxis. So chemotaxis is when chemicals are secreted to attract these macrophages and stuff, or these phagocytes to the area. So again, they are attracted by the chemicals. So here they arrive to the area, and there are bacteria that are present. Adhesion is the sticking of those receptors to those molecules on the surface of the bacteria. So the bacteria sticks to the surface of the phagocyte, and then the phagocyte is going to engulf it and form a phagosome. So this is going to be in the other picture. So we have adhesion here of the bacteria, and then we have engulfment. It just pulls it into a vacuole, right? Makes a vacuole with all the bacteria that's stuck to the cell membrane here. And so now it has a phagosome here. And with its genetic material and stuff, and this is the endoplasmic reticulum and the Golgi apparatus, which makes proteins, and enzymes are proteins, so it's going to manufacture a bunch of enzymes, and they're going to put them in the vacuoles, and those vacuoles are going to merge with the vacuole, the phagosome that has the bacteria, and then you get a phagolysosome formation, and those little enzymes are going to kill and destroy the bacterial cells, and then you just have little molecular debris. It's just down to its little building blocks, and it basically kind of puts it out, if you will. It just excretes the little debris, and there you go. Here's some building blocks and molecules and stuff, and then we eliminate it. Potentially reuse things that are useful. So let's talk about some few inflammatory mediators and cytokines. So some of the non-specific mediators of inflammation and immunity. Again, so these are some of the chemicals that can cause chemotaxis and stuff. So we have tumor necrosis factor, and I'll let you read more detail about each one. I want you to know these, at least be familiar with these names. We have interferons, and there's several of them. Like interferon gamma is a big one. We have interleukins, especially interleukin 1. There are also a bunch of different interleukins, and they have numbers that follow each one. So interleukin 1 is a big mediator of inflammation and immunity. The cytokines that can activate specific immune reactions are interferon gamma. So again, that's a specific interferon, and it's a T cell derived mediator whose function is to activate macrophages. It can also promote the differentiation of T cells and B cells, activate neutrophils, and stimulate diapedesis and stuff. Another one is interleukin 5. The vasoactive mediators, so they act on your vessels. We have histamine, serotonin, and bradykinin. So histamine is produced by mast cells and basophils and can cause vasodilation and increase vascular permeability and mucus production. So think of all the allergy symptoms, signs and symptoms. Serotonin, which is also a happy molecule, right, is produced by platelets and intestinal cells. It can cause smooth muscle contraction and stuff. And bradykinins are vasoactive amines from the blood or tissues, and stimulates smooth muscle contraction also, and increases vascular permeability. So you get redness and swelling and all of that with these guys. The cytokines that regulate lymphocyte growth and activation. So you have interleukin 2 and macrophage colony stimulating factor. And other miscellaneous ones, we have prostaglandins. You can thank prostaglandins for like sinus headaches and stuff like that. And depending on where they're located and what type of prostaglandin it is, it can have various effects. But they pretty much are really powerful at stimulating pain and inflammation. Leukotrienes they can stimulate the contraction of smooth muscle and enhance vascular permeability also. But they are responsible for like constriction of airways and allergy reactions. And then your platelet activating factors, they're released from basophils and cause aggregation of platelets and clotting and stuff like that. So these are all of those. And so we mentioned diapedesis. So that is a process. That's process by which these cells can leave the bloodstream and enter tissue. So the capillaries, this is at a capillary. Uh, when there is an inflammation in the area, there's an increased permeability. So there's an increased space between the cells, and these white cells can literally squeeze through these spaces and follow the chemotactic factors to the area of infection. So this is an electron micrograph of neutrophils. Here's the border of the blood vessels, and these guys are squeezing through and leaving and going into the tissues. All right, so let's talk fever. Fever is a body temperature. Normally, your body temperature is 37 Celsius or 98.6. If you have a low-grade fever, it would be 37.7 to 38.3 or 100 to 101 Fahrenheit. And a high-grade fever is 40 Celsius to 41.4 Celsius or 104 to 106. Beyond 106, it's like about deadly. It can cook your brain and stuff like that. So those are the various fever. If your body temperature is lower than 37 Celsius or 98.6, you may have thyroid issues, and it can be a little bit harder to detect a low-grade fever in a patient that normally has lower body temperature. Pyrogens are substances that reset the hypothalamic thermostat to a higher setting, causes you to run fever. So pyro for fire. Okay, so they induce fire or induce fever or heat. Right? Exogenous pyrogens are products of infectious agents such as viruses, bacteria, protozoans, fungi, endotoxins, blood products, vaccines, or injectable solutions. So they came from outside of the body and they're inducing fever in the body. An endogenous pyrogen are these are chemicals that are liberated or released by monocytes, neutrophils, and macrophages, so they come directly from your immune system. While those cells are doing phagocytosis, and there are molecules such as interleukin-1 and tumor necrosis factors. So if these guys are released, you'll be running fever. The benefits of fever is that it impedes the nutrition of bacteria by reducing the availability of iron. So fever causes you to hide the iron from the bacteria. The bacteria need the iron to grow. It also increases metabolism. It stimulates immune reactions so that your immune system works faster. And it is naturally protective. It's a naturally protective physiological process. So then the question becomes, should you control the fever or should you just let it be? So I would advocate, if possible, let that fever be. But each can make its own decision. Antimicrobial proteins, interferons. So interferons is the interferon is a small protein that's produced naturally by certain white cells and tissue cells, often in response to viruses. So interferon alpha and beta are produced by lymphocytes, fibroblasts, and macrophages, and interferon gamma are produced by T cells, again, usually in response to viruses. They are not microbe specific. They are a valuable treatment for a number of viral infections. They are kind of expensive too. The biological activities of interferons are they can bind the cell surfaces and induce changes in genetic expression. So the exact results vary. So basically they can turn certain genes on and maybe others off to facilitate you fighting off the infection. All three interferons can inhibit the expression of cancer genes and have tumor suppressor effects. And interferon alpha and beta stimulate phagocytes, and interferon gamma is the immune regulator of macrophages and B cells and T cells. So let's talk about complements. So named for its property of complementing the immune reactions, it is consists of 26 blood proteins that work together to destroy bacteria and certain viruses. These chemicals do a cascade reaction, which is basically a sequential physiological response. Think of a domino effect. Like one affects the next, which affects the next, which affects the next, which affects the next, and eventually you have the effect. The first substance in a chemical series activates the next substance, so the domino effect, which activates the next, and so on. There are 3 different pathways in the complement cascades, and they're distinguished by how they are activated, but they all have the same end result. So those 3 pathways are the classical, the alternative, and the lectin pathways. And so this is an illustration of what complement do. So for example, you have complement C3, which is hydrolyzed or split into C3A and C3B. C3B is going to attach to C5, which splits it into C5A and C5B, and then C5B, C6, C7, C8, and C9 work together to make the membrane attack complex, which basically pokes a hole in the bacterial cell membrane, and it causes the cell to swell and burst. And so you can see it here on the electron micrograph. So it basically blows cells up. Kind of cool. So antimicrobial peptides are short proteins that are 15 to 20 amino acids long. They are able to insert themselves into prokaryotic membranes to kill microbes. They are called defensins, marganins, and protegrins. They also have an effect on other actions of non-specific and specific immunity, and researchers are looking into ways to turn these antimicrobial peptides into therapeutic drugs to help you fight off infections and stuff like that. So give me one chemical mediator of inflammation or a cytokine, any of them, just list it there. And we're going to look at another case. So this is a missionary couple that lives in West Africa, and they brought their 4-year-old son to the office of their physician on the second day of a visit home in Minnesota. The boy had a mild episode of diarrhea about 7 days earlier, and he would not eat. He seemed to recover, but the mother noticed that the boy was having trouble walking the previous night and had seemed to have trouble dressing himself and walking that day. On examination, the patient had no significant fever. He had normal bowel sounds. His chest, ear, and eyes were clear. He had no rebound tenderness in the abdomen, which means no appendicitis. The physician noted that the child had poor muscular reflexes in his arms and legs. The child also was a bit lethargic and seemed confused. The family lives in an isolated village in Africa. The mother opposes vaccination on personal grounds, so the child has only had the initial series of DPT shots and no other typical vaccines. The village where they live has many problems with parasites and insect-borne fevers, including dengue. The urine, stool, and blood samples were collected for analysis, and the results indicated that this child had polio, which we do have a vaccine for. So, okay. How could he have gotten polio? More than likely, he was exposed to an asymptomatic carrier, which is why I have the little star here. So in the village, because the isolated villages in Africa, more than likely, it's rare that the children are vaccinated, even though they do vaccination campaigns and stuff like that. Okay, so let's talk a little bit about carrier state. So you have asymptomatic carriers are infected, but do not show symptoms of disease. So, well, of course, if you don't show symptoms of disease, you're not going to get treatment, and you don't know that you can spread it to other people. This is very, very common with gonorrhea, genital herpes, and HPV. And so you just don't know that the person has it, and if you have unprotected sex, for example, you can have it. You can get infected. Incubating carriers, they can spread the infectious agent during the incubation period of the phase. So again, they're in that, like we were talking about coronavirus, they're in that phase between being exposed and developing the first signs and symptoms. And so the microbes are in there, they can multiply, viruses in there multiplying, and if they cough, if it's respiratory or whatever, or whichever way, they can spread it, okay? And then we have convalescent carriers. So those are the ones that are recuperating, but in certain instances, they can still spread the disease. So hepatitis A is one where the convalescent carrier can spread the disease. And then we have chronic carriers, or individuals who shelter the infectious agent for a long period after recovery because of the latency of the infection agent. So tuberculosis and typhoid fever are two of those where you can have chronic carriers and they're not sick, right? They're fine. And then you have passive carriers, are medical and dental personnel who constantly handle patients and patient materials and stuff, and are heavily contaminated with patient secretions in blood, and they risk picking up pathogens and then mechanically or accidentally transferring them to another patient. So again, this is where it's very important for you to like wash your hands and all of that, but then even think your stethoscope or other things probably should be wiped down and cleaned between patients too. Okay, so then let's talk about reservoirs. So if you think about a reservoir, like a water reservoir is something to contain water. Okay, so a reservoir in microbiology is either a living or non-living, if you will, containment unit or containment entity for that specific disease. So in our reservoirs, we have living reservoirs such as, so we have animals other than humans and arthropods. So you have mammals, birds, reptiles, etc. They just carry the disease. So think rats, for example. Then you can have humans. You have humans that are actively ill or humans that are carriers. Again, so in our case, it would have been a carrier or silent carrier that infected that child. And then you have arthropods, so they're biological vectors such as like your fleas that are biting, ticks that are biting, mosquitoes that are biting, and all of that. And so like malaria is a good example of that, and with a mosquito in malaria. That would be an arthropod vector. And then the non-living reservoirs are soil, water, and air in the built environment. So the fact that we have air conditioning systems and things that circulate air that can be contaminated. So some pathogens such as the TB bacteria can survive for long periods of times in non-living reservoirs such as in the air and stuff, and it can be transmitted to humans when they come in contact with contaminated soil, water, or air. So when we were looking at the brain-eating amoeba, so it lives in water and stuff, and so that would be its reservoir. Okay, so I want you to list me one zoonotic infection. So a zoonotic infection is an infection that animals can have. It can spread to humans. So do you know one? Put it here. I'm fixing to show you a bunch. Okay, so here are some examples of common zoonotic infections. So we have rabies, yellow fever, several viral fevers. We have hantavirus that can be found in rodents, influenza in chicken, birds, and swine, West Nile virus. So those are all the viruses. The bacteria, you have Rocky Mountain spotted fever, you have leptospirosis, you have anthrax, you have the plague, and then in the miscellaneous, you have ringworm, toxoplasmosis, trichomoniasis, and all of that. Tapeworms, all kinds of stuff. So how do we get, so acquisition and transmission of infectious agents, how do we get sick with these? A disease that's communicable is a disease in which an infected host can transmit the infectious agent to another host and establish the infection in that host. Infectious is synonymous with communicable, so it can be communicated or spread from person to person. The flu, influenza, is a communicable disease. Contagious, a disease that's contagious is a disease that is highly communicable, especially through direct contact. So again, influenza and measles are communicable, but they are highly contagious, so they're very easy to spread. Hansen's disease or leprosy is only weakly communicable. So you have to actually have physical touch with like infected parts of the person in order for it to spread. Non-communicable is an infectious disease that does not arise through transmission of the infectious agent from host to host. So examples of that would be compromised persons that are a compromised person that's invaded by its own microbiota. So that yeast infection, for example, also would have been in the patient had antibiotic would be a kind of a non-communicable. You're not going to spread it. An individual has accidental contact with a microbe that exists in a non-living reservoir. That would be you don't spread it. So that girl that got brain-eating amoeba, Naegleria fowleri there, she's not going to spread it to other people, okay? And the infected person does not become a source of disease to others. So that's the idea of non-communicable. So sometimes you have patients that would have a flu and then develop pneumonia from an endogenous bacteria, and these guys are actually the disease is non-communicable. Once they've recovered from the flu, that pneumonia, they have that bacterial pneumonia, is not something that will spread. So patterns of transmission in communicable diseases. Vertical is always going to be mother to child through the process of carrying a child and delivering a child. So, for example, if a mother has HIV, the child could have HIV, right? If she has it while she's pregnant. Okay. And then horizontal is really how disease is spread. It's from friend to friend, or between family members. And so in that scenario, you have direct contact. So kissing somebody that has mono Epstein-Barr virus, and then you can get mono, right? So touching, kissing, sex, droplet contact, which is like close contact, talking, playing and stuff with somebody. So colds and chickenpox are droplet contact. And then parenteral is transmission via intentional or unintentional injection into deeper tissue. So using needles, knives, branches, broken glass, etc. So that is direct contact transmission. And then, of course, indirect is the infectious agent has to pass from an infected host to an intermediate object or whatever, and then from then to another host. So that's the typical doorknob, that's fomite. So inanimate objects, phones are a big one too, faucets, handles and stuff like that, doorknobs. Right. The vehicle is a natural non-living material that can transmit infectious agents. So we have air, water, soil, and food. And again, the oral fecal route can use either vehicles or fomites, a fecal carrier with inadequate personal hygiene. Remember the whole hepatitis A thing in this area? There you go. People not washing their hands, contaminate your food. You eat your food, you get hepatitis A. There you go. And vector transmission then is a mechanical vector would be like the reason we don't like flies. So they land on dog poop and then they go land on your food, right? And the little paws there, or little legs, not really paws, the little legs carry the bacteria from the poop to whatever food they land on. Okay, so that would be a mechanical vector. And a biological vector would be like the mosquito bite, so not where the parasite or the virus or whatever is part of the life cycle there. And it goes inside the mosquito, and then the mosquito bites another host, and it goes into another host. And we're almost done. Epidemiology is the study of disease in populations. So we've been getting these daily reports, and some of the stuff they're reporting are epidemiological statistics and stuff. So epidemiology is the study of frequency and distribution of disease and other health-related factors in defined populations. It involves many disciplines, so microbiology, anatomy, physiology, immunology, medicine, psychology, sociology, ecology, and statistics. It considers all forms of disease. So we have statistics, epidemiological statistics on heart disease, cancer, drug addiction, mental illness, and of course infectious diseases too. The statistics of concern to the epidemiologists are rates of disease with regard to sex, race, or geographical region. The mortality rate measures the total number of deaths in a population due to a certain disease. The overall death rate from infectious disease has dropped, although the number of persons afflicted with infectious diseases has remained high. That is the morbidity rate. So the morbidity is how many people are sick or have the disease. Mortality is how many people die of the disease. The index case is usually the first patient found in a epidemiological investigation. It may not be the first case of the disease, but it's the first one that's been brought to the attention of the officials. So, endemic is an infectious disease that exhibits a relatively steady frequency over a long period of time in a particular geographical locale. So, for example, in this area, we are endemic for histoplasmosis. Histoplasma is endemic in this area. There's always some cases. It's out here, it's out in nature, and so cases are going to pop up kind of pretty steady throughout the years, over the years. Sporadic is an occasional cases are reported at irregular intervals at random locales, so there's no pattern to it. And an epidemic is when statistics indicate that the prevalence of an endemic or sporadic disease is increasing beyond what is expected in a population. So you can see the numbers go up. So, for example, when you see we've had foods that are contaminated, like peanut butter that was contaminated, and you had an epidemic of, I think it was salmonella or something like that, in certain area, and due to the consumption of specific food. Example. So a time period is not defined for the epidemic. It can be measured in hours to years. The exact percentage of increase needed before an outbreak is qualified as an epidemic is also not defined. So how many cases consider an epidemic? What kind of increase? We just haven't decided. It's all of a sudden like, oh, looks like more than normal, so now we have an epidemic. And then a pandemic is an epidemic that spreads across continents, and that is what we're dealing with the coronavirus. So when it was located in China, it was an epidemic in China. As soon as it started crossing continents, it became a pandemic. So which of the following terms describes the total number of persons afflicted with a particular infectious disease? Answer that question. And then latency and sequelae. Latency is the dormant state of microbes in certain chronic infectious diseases. So viral latency, the viruses that have viral latency are herpes simplex. It never goes away. Herpes zoster never goes away. If you've had chickenpox, you'll get shingles, or you can get shingles. Hepatitis B, AIDS or HIV basically, and Epstein-Barr, which causes mono. For bacterial and protozoan, the ones that have latency are syphilis, typhoid fever, tuberculosis, and malaria. So they're hard to treat and don't really go away either. And then sequelae are the long-term or permanent damage to tissues or organs that are caused by infectious diseases. Meningitis can cause deafness. Strep throat can cause rheumatic heart disease. Lyme disease can cause arthritis. Polio can cause paralysis, like in our case. And we're even seeing, like for coronavirus, there are some kids that are having cardiovascular problems as a sequelae of coronavirus infections. Okay, and that is the last slide. And I hope you took some breaks because I'm tired. Anyway, I will see you in the next lecture. Thank you.